Bismillah Rahman Rahim. Welcome back, friends. Welcome to another video lecture by Kami Biology. So we talked about hypersensitivity introduction, and we talked about type 1 and type 2 hypersensitivity. Now in this video lecture we are going to talk about type 3 hypersensitivity. So let me write type 3 hypersensitivity. If you guys remember in the previous video lecture, in type 2 hypersensitivity, we talked that it involves antigen and antibody. It involves antigen antibody complex due to which agglutination occur. Now similarly in this type 3 the antigen antibody are also occur, and in this type 3 agglutination process can also happen. Okay, in the previous video lecture, if you remember, we talked that such antibody are activated against the RBC cells. Means on the surface of the RBC there are some antigen, so such antibodies are activated against these RBCs. In the previous video lecture, if you remember, we talked about for the understanding of type 2 hypersensitivity, we first talked the ABO blood group system. First we try our best to understand on this ABO blood group system. Now in this video lecture, what we are going to talk about, we are going to talk about type 3 hypersensitivity. But before understanding this type 3 hypersensitivity, we need to understand about the complement system, because in this type 3 hypersensitivity it is related to the complement system. In type 2 hypersensitivity it is related to the blood group system. So that's why in the type 2 hypersensitivity we also a little bit talked to blood group system. Okay, so don't worry about the complement system. Inshallah, in some other video lecture we will explain the complement system in a detail, but here I will just clear your concept. We will just talk the basics, the basis of the complement system. So let's first of all a little bit explain the complement system, that what is actually the complement system and what kind of the proteins there are, how many proteins are involved. In some books they say that there are 20 proteins. In some books they say that there are 30 proteins are involved. Okay, but usually they contain 20 proteins. First of all, what actually happened, let's suppose if we first of all let me explain a little bit about the complement system. Let me a little bit clear your concept on complement system, that what actually happened in the complement system. Now if we talked about the complement system, so let me write complement system. Complement system. Now if we talked about the complement system, so one thing that I must tell you, inside the complement system, as I told you, that there are 20 or 30 proteins are involved. So in each of the protein, such protein have two domains. How many? They have two domains. So let's suppose we have the proteins like that. Let me draw with the green marker. Let's suppose we have proteins like that. Means this is what this is. Let's suppose the protein. So they contain what? They contain two subunits, as you are watching. This is the small subunit and this is the large subunit. Whenever the complement systems are activated, the complement system are not usually activated. For the activation of the complement system, first of all, IgG antibody are involved. We have IgG antibody. So let's suppose we have what? We have IgG antibody first of all involved. Now these are what? Let's suppose these are the IgG antibodies. In this type 2 hypersensitivity, we will also fully focus on the IgG antibody. And this type 2 hypersensitivity, the IgG antibody are also involved. So first of all, what actually happened, this IgG antibody, let's suppose we have a bacterial cell also. Okay, we have what? We have the bacterial cell also. So let's suppose this is our bacterial cell. First of all, what actually happened, two IgG antibody will sit here. Not only two, two antibody. I think two antibody will sit here. So these two antibody are what? These two antibody are IgG antibody. Okay, so they will sit on the bacterial cell, and once they sit on the bacterial cell, then they will call upon the complement system, complement proteins. So the first complement proteins we have CQ1. Okay, C1Q, I think. Yeah, C1Q protein. This C1Q protein have C1R and C1S. I think just like that subunits they have. So they will, don't worry about this, I will explain it, and I will, means clearly explain it. But maybe here I do a little bit mistake. So don't worry about this, inshallah I will explain it in much more detail. Let's suppose there are what? These are the FC portion as I draw. At the FC portion, there are, means proteins are involved. So these are what? Let's suppose these are the C1q proteins are involved. Due to this C1q proteins, once they are bind to that area, this is the C1q proteins. What? This is the C1q protein. Once the C1q proteins are binded to here, then they will provide some signal to other complement system, other complement proteins, sorry. So other complement proteins are also activated. So once the other complement proteins are activated, then before activation, let's suppose we have another complement system activated, which we call it C2A. Okay, so we have what? We have C2A. So this is C2A. Sorry, this is C2 protein. Now this C2 protein have two subunit. One is the large subunit and the other is the small subunit. Except this C2, some of the proteins means C4, C5, C3 having two subunit, but the large subunit is B and the small subunit is A. But only in C2 case they have large subunit A and small subunit B. This is very important concept. Try to understand it. So this protein is activated and they will go there. They will, this protein will go there. It is very small protein, but actually I draw here a very large for your understanding. And once they go there, so they will be dissociated, or they will be, breaked into two parts. So now this is break into two parts. So after it break into two parts, the first portion is released and the second portion is attached. So here, this is what? This is the C2A protein. So C2A protein will attach here. Similarly, we have C3 proteins are activated. C3 proteins are activated, and again C3 protein have two subunit. One is, now in this C3 protein case, they have large B subunit and small A subunit. So they will again come and sit here. So let's suppose there what? There are the, another proteins connected, and we call that C3b protein, and C3a is released. Similarly, C4a protein are bind here, and so on. The proteins are banded here, and due to these proteins, let's suppose there are the proteins are banded and banded and banded, and what they are causing, they are creating the holes as you are watching. So a holes is created. Okay, these are what? These are the proteins, means C3a, C2b, C5, C6, C7. So almost 20 proteins are binded to here, and these as such protein can create the holes. And due to these holes, the ions and the water molecule which are present outside the cell, these ions will internalize, due to which the cell will swell up and burst, and finally the bacteria cell will die. Okay, so this is the complement system through which the bacteria are die. Okay, so this is how the complement system are activated. It is not so clear as I draw. It is not so clear, but just try to understand that the complement system are involved, or complement system process will be happen just like that. Inshallah, I will explain much more clear in some other video lecture the complement system. Now let's talk about the type 3 hypersensitivity. Type 3 hypersensitivity also have the same process, but what actually happen? This type 3 hypersensitivity is also called antibody mediated reaction. So let me write. It is also called what? Antibody mediated reaction. So we also called it antibody mediated reaction. Now, why we call it antibody mediated reaction? Because in this case, antibodies are involved, and usually IgG antibodies are involved. Okay, first of all, what actually happened? Let's suppose our body is exposed to an allergen. This allergen could be a pathogen. This allergen could be useful, harmful, dandruff, animal tissue, any kind of things that will just trigger the immunity, or that will just trigger the immune system. What actually happen once our body is exposed to such allergen, then let's suppose our IgG antibody is activated. So this is what, this is the IgG antibody which are activated. So once IgG antibodies are activated, due to what? Due to this means allergen. Now this is what? This is the allergen molecule. Now once IgG antibodies are secreted or activated due to this, due to this what? Due to this allergen, then what actually happen? They will make antigen antibody complex. We also call it immunocomplex as well. So they can make antigen antibody complex. What actually happened after that? After that, this antigen antibody complex, here I draw just two antibody, but actually it is, as it is hypersensitivity reaction, so it means a lot of IgG antibodies are activated, hyper, so increased, a lot of IgG are increased. So let's suppose these are a lot of antibodies are increased, IgG antibodies are increased, and they will, make a complex antigen antibody complex. Each complex will settle, or we can say each complex will load gain. So let me write load gain. What does it mean? It means that they will stay for a while on what? They will, settle and stay for a while on, let's suppose these are what? These are the tissue cells. These are our tissue, let's suppose. Tissue cells. Okay? These are what? These are the tissue cells. These may be the, tissue cells of our vessels, blood vessel tissue cells, lungs tissue cells, or we can say these tissue cells are, these are, vessels. Let me a little bit says that which kind of tissue these are. So these tissue will be vessel, blood vessel. Okay, what they are? Blood vessels, let's suppose. They may be blood vessels. They may be, lungs tissue. They may be heart, kidney one, skin. Okay, and joints. So these are the tissues. Such antigen antibody will loading on the surface of each tissue. Each tissue will be blood vessel tissue, lungs tissue, heart, kidney, skin, joints. Once, let's suppose these are what? These are the antibody, antigen antibody complex. These are what? These are the antigen antibody complex. Let me draw the antigen here. No, these are what? These are the antigen antibody complex. So antigen antibody complex will settle or loading on the basement area of what? On the basement area of each tissue cells. Once it is loading on that area for a small period of time, then due to this way, they will activate the complement system. So then what actually happen? The complement systems will be activated. Due to this complement systems, at their Fc portion, as you are watching, the antibody, at the Fc portion of the antibody, there are the complement proteins are connected. So a large subunit of the complement system protein. The complement system protein, their large subunit will be connected to this Fc portion of the antigen antibody complex, and their small subunit, let's suppose their small subunit, these green ones are what? These green ones, the small subunit of these complement system. these small subunit act as a chemotactic factor. They will call upon some other cells. Okay, so they will what? They will call upon some other cells, and these other cells we have what? We have, dendritic cells. Tick. We have macrophages. We have neutrophils. We have monocytes. Usually we call that phagocyte cells. So they call upon the phagocytes, and inside the phagocyte they will call upon dendritic, macrophages, neutrophils, monocytes. So these cells are get activated. And what actually they do? Once these cells are get activated, so then each of the cell will cause the damages of each tissue. So each tissue will be what? Each tissue will be damaged. So these epithelial tissue, okay, these epithelial tissue will be destroyed. Let's suppose these epithelial tissue are the blood vessels. Okay, means each epithelial tissue are what? Each epithelial tissue are the blood vessel. So when it destroy the blood vessel tissue, so then they will cause the inflammation of the blood vessel, which we call it vasculitis. What? So whenever they will do that, so we call it vasculitis. So the inflammation of the blood vessels due to this dendritic, neutrophils and monocyte, due to this fragments of the complement system, so then we call it vasculitis. When each epithelial tissues are the lungs, so then they will cause inflammation inside the lungs, which we call it bronchitis, or sometimes we call it pleuritis. Okay, so let me write. So these are what? These are the bronchitis or pleuritis. If these epithelial tissue are destroyed, and such epithelial tissue are the part of heart, so then this may cause what? Inflammation inside the heart, which we call a myocarditis. So let me write myocarditis. If these are the kidney tissue, means these are the epithelial tissue of the kidney which are destroyed by each of the cell through this hypersensitivity 3, so then we call that what? We call that glomerulonephritis. If these are the epithelial tissue of what? If these are the epithelial tissue of these joints, so then they will cause arthritis. So they will cause inflammation of the joints, which we call it arthritis. If these are the tissue of the skin, epithelial tissue of the skin, and if the skin epithelial tissue are destroyed by such phenomena, by such complement system, by such macrophages, all these things are happen in type 3 hypersensitivity, then we call that dermatitis. Inflammation of the skin, we call it dermatitis. Okay, so what actually happened? These fragments, as you are watching, these fragments are actually, sometimes they are acting as a chemotactic factor, or sometimes they will produce histamine, and due to histamine they will call upon each of the cell. So that's all about the hypersensitivity type 3. Inshallah, in the next video lecture we are going to type 4 hypersensitivity. So stay tuned and continue to watch the video. Thank you.